Hey, boys and girls. So when we take a look at the Punnett Square practice, let me talk about a couple things real quick. One of them is when we have to look at the terminology, like homozygous yellow plants. You can see that we're using Y's, the letter Y, because yellow is dominant to green. And so it's capital Y means homozygous, homo being the same. And that shows me that Y is dominant to green. And so they both code for seed or coat color, and so that's why we use the capital letter Y. And so you don't need to color the alleles themselves. Alleles are the different form of the same gene. Each parent passes off one. And so this is one right here, offspring. It's capital Y, capital Y. And here's the other one, lowercase y, just in this column right here. You don't need to make them blue or red. I just did that to show you where they came from. And so the outcome of that is we simply just bring them down into the rows and columns, those Ys. The blue Ys come down, and the red Ys just move over in their respective rows. The genotype then, for each question, you're going to list genotype and phenotype ratio. So genotype, the genetic code, 100% of them all have the exact same. They all came out to be capital Y, lowercase y, heterozygous. And then the phenotype, the physical appearance, the phenotype, since they all have one capital Y, they're all yellow. So you're going to do that for every question. Do your Punnett square, number it, show me the genotype percentage and the phenotype percentage. Now let's take a look at question 5, for example, because some students were confused about what to do when the letters are not given to me. So for example, when we are looking at a homozygous dominant brown mouse is crossed with a heterozygous brown mouse, tans recessive. So given that's the case, it will look something like this. Here we have the Punnett square. Let's go ahead and make that a little cleaner. That was not nice. So here we have the Punnett square, and our Punnett square is going to look something like this. And we have brown, homozygous dominant brown. Since brown is the dominant trait, I'm going to use a capital B. Homozygous means the same, so it's one capital B, another capital B. There we have that. Since it's with a heterozygous, I already know that's a capital B, hetero being different, and lowercase b. There we have it. So do your Punnett square, your capital B's go over. And then your capital B from the top comes down. Your lowercase b goes in the box. Same thing over here. Your capital B from up top comes down. Your lowercase b goes over. Now for the outcome here, my genotypes, you can see I have two types. Up here, my genotype, I have half of them, or 50%, were capital B. They were all dominant, capital B. And the other half, or 50%, came out to be heterozygous, capital B, lowercase b. The other outcome for my phenotype, if you take a look here, all of them, 100%, have one capital B, which means they're all going to look brown. And that's how we do this type of Punnett square. We set that up there. So I think I ran out of space there, but I think you get the idea. So list the genotype and phenotype for all of them. Make sure you use the letter of what's dominant. So for example, for number 6, it says 2 white rabbits are crossed, brown's recessive. So for that, you're going to use W as your letter because white's dominant to brown. Same thing. 2 heterozygous red flowers, white recessive. So you're going to use R for red. Then likewise for number 8, try that one. Dominant brown mouse, capital B. Okay? Hopefully that helps. If you have questions, let me know.